Hi friends, welcome back to my channel. In this video, we will delve into the exciting world of advanced molecular cloning techniques. We will explore the fundamental concepts, various methods, and the recent advancements in the powerful field used to manipulate DNA molecules. By the end of this session, you will gain a comprehensive understanding of how to clone genes, modify them, and introduce them into living organisms for functional studies. So let's see what is molecular cloning. Molecular cloning is a fundamental technique in the molecular biology that allows scientists to isolate, amplify, and manipulate specific DNA fragments. It essentially involves taking a piece of DNA, the insert, and incorporating it into a carrier molecule called the vector. This recombinant DNA molecule can then be introduced into a host organism, typically a bacteria, for replication and generating numerous copies of the desired DNA fragment. This process has revolutionized our ability to study genes, understand their function, and develop novel therapeutic strategies. So let's see what are the applications of molecular cloning. Molecular cloning has a wide range of applications in various scientific fields. It is a cornerstone of functional genomics, enabling the investigation of gene expression patterns and regulatory mechanisms. By cloning genes, scientists can produce large quantities of recombinant proteins for research purposes such as enzyme characterization or drug development. Furthermore, the molecular cloning plays a crucial role in gene editing, where the specific DNA sequence can be modified to study the gene function or develop gene therapy strategies for treating genetic diseases. And additionally, the DNA forensics heavily relies on cloning techniques to analyze the DNA fingerprints and to identify individuals. What are the essential components of molecular cloning? Successful molecular cloning relies on several key components. The DNA insert is the target DNA fragment we want to clone. Vectors such as plasmids, the bacterial artificial chromosomes, or the artificial yeast chromosome, these act as a carrier for insert and facilitate the replication within the host organism. Restriction enzymes are molecular scissors that cut DNA at specific recognition sequences, generating fragments with complementary overhangs, or it is called as the sticky ends or the blunt ends. DNA ligase then acts as a molecular glue, stitching together the insert and vector DNA to form a recombinant molecule. The choice of the host organism depends on the desired application. For example, bacteria like E. coli are commonly used due to their rapid growth and ease of manipulation. Finally, the competent cells are the host cells that have been treated to make it permeable to foreign DNA, allowing the uptake of recombinant DNA molecules. So these are some of the basic terms that is used in the molecular cloning. So let's see each one of the cloning techniques in detail. So first one is the restriction enzyme cloning. This is also known as the traditional cloning method. So restriction enzyme cloning remains as a popular and robust method for molecular cloning. And here are the breakdown of essential steps involved in restriction cloning. The first step is to prepare the DNA fragments. So we begin by isolating the DNA fragments of interest, typically through PCR or a genomic DNA extraction. And this fragment is then digested with specific restriction enzymes chosen based on the recognition sequence present within the fragment. The restriction enzyme cleave the DNA at specific short palindromic sequences, generating fragments with either sticky or blunt ends. Next comes the vector preparation. The vector, which acts as a carrier molecule for our insert, also need to be prepared. So we linearize the vector using same restriction enzyme employed for the insert. So the vector and the insert need to be restricted or the fragment needs to be cut based using same enzyme, and this generates complementary ends on both vector and the insert, facilitating their ligation. An additional step involving dephosphorylation of the vector ends is often performed, and this is actually to prevent the self-ligation. Next step is the ligation. Once the insert and the vectors are prepared, they are combined in a reaction mixture containing DNA ligase, an enzyme that acts as a molecular glue. DNA ligase covalently joins the phosphodiester bonds between the insert and vector fragments, forming a recombinant DNA molecule. So once the ligated product is ready, this need to be transformed. So next step is the transformation. The recombinant DNA molecule needs to be introduced into a suitable host organism for replication, and this process is called transformation, which involves making the host cell competent to take up the foreign DNA. So competent cells can be generated through various methods such as heat shock or treatment with chemicals, and this recombinant DNA is then introduced into the competent cells, and a small portion of cells will successfully take up the plasmid. Once it is successfully transformed, we have to select between the bacterial cells with the recombinant plasmid, and one without that need to be screened out. So for selection, we need to identify and select the cells that have been successfully transformed with the recombinant DNA molecules. And selection markers incorporated within the vector play a crucial role in these steps because that can be used as a markers for selecting these. Commonly used selection markers include antibiotic resistant genes. For example, if the vector contains an ampicillin resistant gene, only the transformed cells will grow in the media containing antibiotic. The non-transformed cells will be unable to grow because of the absence of the resistant marker present in the plasmid. So this is one way of selecting the recombinants. The restriction enzyme cloning offers a reliable and well-established approach for cloning DNA fragments. However, the requirement of complete restriction enzyme sites within the both insert and the vector can be sometimes limiting its flexibility. So there is an alternative to that is known as the PCR based cloning. So PCR based cloning offers a powerful alternative to the traditional restriction enzyme cloning. And the polymerase chain reaction, it's a technique that exponentially amplify the specific DNA fragments. In PCR-based cloning, the primers containing the restriction enzyme recognition sites are designed to flank the target region sequence, and the PCR product and the vector are digested with compatible restriction enzyme, generating fragments with complementary overhangs. So it is basically introducing the restriction sites through PCR by designing the primers to have those sites in the primer, and during the amplification, this will introduce a restriction site to the PCR product. So we are not dependent on the restriction sites in the sequence. We are actually introducing the required restriction site to the insert by PCR. Another method is called as the recombinational cloning. Recombinational cloning is a site-specific scarless method for DNA cloning. This technique utilizes enzymes like CRE recombinase or FLB recombinase, which mediate recombination between specific region sites, recognition sites called the ATT sites, flagged within the vector and the insert. So these enzymes promote strand exchange between insert and the vector DNA at ATT sites, resulting in the seamless integration of the insert into the vector. Recombinational cloning offers several advantages, including directional cloning, elimination of the need for restriction enzyme, and ligation and generation of scarless vectors without any unwanted sequence introduced during the cloning. So there is no restriction, fragmentation or DNA ligation happening in this. So this actually happens through the recombination. Next method is the seamless cloning. Seamless cloning is an in vitro cloning method that relies on the overlapping ends of the assembly DNA fragments. In this technique, the DNA fragments are designed with complementary overhangs of approximately 15 to 20 base pairs, and the enzymes like T5 exonuclease and Taq polymerase play a crucial role. The T5 exonuclease chews back on the 5' end of the fragments, resulting in single-stranded overhangs with perfect complementarity. Taq polymerase then extends these overhangs, filling any gaps and creating double-stranded DNA molecule. Finally, DNA ligase seals the remaining nicks, resulting in a seamless recombinant molecule without any additional vector sequence. So now we have learned the different methods of cloning. Now let's see how to select suitable vectors for molecular cloning. The choice of vector is critical for successful molecular cloning and depends on several factors. One is the size of the insert. Size of the insert, the size of the DNA fragment you want to clone is primary consideration because the plasmids are most commonly used vectors due to their small size, typically ranging from few thousands to 10,000 of base pair. They are ideal for cloning smaller DNA fragments. However, larger inserts exceeding the capacity of the plasmids, alternate vectors need to be selected. The next is the expression requirements. If your goal is to have cloned gene expressed in a host organism, you will need to ensure that expression vector. We need to use an expression vector. Expression vectors are specifically designed for this purpose, and they contain regulatory elements like promoters which initiate transcription of the gene and terminators which signal the termination of transcription. These regulatory elements ensure efficient expression of cloned gene within the host organism. Now comes the selection markers. Selection markers also play a vital role in identifying and selecting the cells that have been successfully transformed with a recombinant DNA molecule, and these markers are typically genes incorporated within the vector that confers a selectable trait to the transformed cell. The commonly used selection markers are antibiotic resistance gene. If the vector contains an ampicillin resistant gene, for example, only transformed cells that will have the antibiotic marker will be able to grow on that particular medium. Non-transformed cells will be unable to grow due to the presence of antibiotic, and other selection markers include blue-white screening system and the reporter genes. Next comes the cloning strategy. So the specific approach you will take for cloning DNA fragments will influence the features you require in the vector. We have seen that directional cloning. So if the orientation of the insert within the vector is critical, you will need a vector with unique restriction enzyme sites that flank the cloning site. and this allows for the insertion from the fragment in a specific orientation. Next is the multiple cloning sites, and the presence of MCS within the vector provides a convenient region for inserting the DNA fragments using restriction enzymes. Many vectors contain single MCS, while others may have multiple MCS with different restriction enzyme sites. The restriction enzyme sites, the availability of compatible restriction enzyme sites within the vector and the insert is necessary for traditional restriction enzyme cloning. However, some of the advanced cloning techniques may not require these sites as discussed before. So, not all the cloning methods require restriction enzyme sites. Coming to the transformation and selection. Transformation, as mentioned earlier, it is the process of introducing recombinant DNA molecule into the host cells. For efficient transformation, the competent cells that are receptive of foreign DNA uptake is very crucial. The selection markers incorporated within the vector play a crucial role in identifying the cells that have been successfully taken up by the recombinant DNA. Commonly used selection markers include antibiotic resistance genes, blue-white screening system, and the reporter genes. Antibiotic resistant genes allow the transformed cells to grow in the presence of specific antibiotic, while non-transformed cells cannot. The blue-white screening utilizes a system where the vector disrupts the gene responsible for producing blue-colored pigment on the media containing a chromogenic substrate. Only transformed colonies will appear white. The reporter genes encodes easily detectable products such as fluorescent proteins and allowing the identification of transformed cells. Now we have the clone study. How to verify we have the correct insert in that. So to verify the clones, it is essential to ensure that successful incorporation of the desired DNA into the vector. Restriction enzyme digestion of the recombinant plasmid allow for analysis of insert size and orientation within the vector, and the DNA sequencing provide ultimate confirmation of the insert sequence, ensuring it matches with the target DNA. The other option is colony PCR, a rapid screening method where the small amount of culture from each colony is used in the PCR reaction with primers specific to the insert. The presence of amplification product confirms the presence of the insert of the particular clone. So in summary, molecular cloning has become a cornerstone technique in modern microbiology research, and the development of advanced cloning technique has significantly enhanced our ability to manipulate DNA with greater flexibility, efficiency, and accuracy. As the research progresses, we can expect further advancements in the cloning technologies, opening doors to even more exciting discoveries and application in the field of science. Thank you for listening. If you like the video, please press the like button and share it with your friends. Thank you.